In this lecture, we will start considering effects of electric field on fluid flow and mass transfer, and this topic is called Electrohydrodynamics and Electrokinetics. Now, the idea is that till now we have limited our discussion to pressure-driven flow and passive scalars or dyes which or solutes which advect and diffuse due to a pressure-driven flow. But now we are interested in what happens if we apply an electric field and ions. So we have ions or charged particles in the solution. So we will see that the coupling of electric field with fluid flow leads to interesting transport phenomena which is called electrohydrodynamics, and this field of coupling of electric field and fluid flow is called electrohydrodynamics, and that can be used in microfluidic devices to pump fluids and also transport ionic species. So if we can pump fluids by simply applying electric field, we can develop a micro pump, and we will discuss how we can do that. And the motion of ionic species due to applied electric field is widely used for DNA, protein analysis, where DNA molecules of DNA fragments of different sizes, they move at different speeds depending upon their charge and size under the effect of electric field, and that's how you can separate these DNA fragments, and this process is called electrophoresis. So we can see that electric field driven fluid flow and mass transport can have and indeed has a lot of applications in development of lab on a chip devices, because you can pump fluids, you can transport charged molecular species, you can also transport charged particles just by applying electric field. Now, to see what kind of transport phenomena takes place when we have charged species and apply an electric field, let's consider a simple case of a glass capillary. So capillary which is made of glass, and in microfluidics typically we would have a capillary or a channel of thickness of order 10 to 100 micrometers, and in this microchannel or capillary we have filled an electrolyte solution . Okay, for example, sodium chloride. Okay, so we have sodium which is sodium ions which are positively charged, and chloride ions which are negatively charged. Now what happens is that if you have an electrolyte solution in this channel, then firstly, when the glass surface comes in contact with water, it acquires a negative charge on the surface, and that's because glass has these silanol groups, SiOH groups, immobilized on its surface, and this group is actually acidic. That means if you bring it in contact with water, it would donate a proton and become negatively charged. So these silanol groups are immobilized on the surface. Okay, so they cannot move But what they do is that because you have this negative charge on the glass surface, it will attract positive ions from the electrolyte solution close to the surface. So that means if you have sodium chloride solution in the capillary, you would have more number of positive ions close to the negatively charged wall, and that's because these positive and negative ions in the solution are mobile, but those immobilized on the surface cannot move. Now what happens is that close to the surface, if we zoom in, we see that near the surface, it's not the case that you just have all positive charges. There are some negative charges, but on average you have more number of positive charges near the surface, and this is a diffusive interface because these molecules are also undergoing random motion due to due to random thermal fluctuations or Brownian motion. So effectively we see that close to the wall you have more number of positive ions, and as you go further from the channel wall, eventually you have the same number of positive and negative ions. So the thickness of this diffusive interface over which you have more number of positive ions than negative ions is usually very small. So that's usually of the order of 10 nanometers or even smaller. And this structure, so you have negative ions and more number of positive ions close to the wall, is called the electric double layer or electrical double layer, which we will call as EDL. So the EDL thickness is usually of the order 10 nanometers or even smaller for electrolyte concentrations or electrolyte ionic strength of order 1 millimolar or larger. Okay, so now this is the case when even if you don't apply electric field, this is the case how the charge we distribute near a charged surface. So charges in the electrolyte we distribute near the charged surfaces, and outside this EDL we have local electroneutrality. That means you have locally same number of positive and negative ions outside this thin region of order 10 nanometers or smaller. Now if we apply an electric field along the axial direction, let's say something like this, then what happens is that these positive ions near the wall, they start migrating under the effect of the electric field, and while they do so, they drag the fluid along with itself. Okay, and that leads to fluid flow. In other words, you can think that you have this free charge inside this fluid, and when you apply an electric field, there's a body force on this fluid because you have these charges and they couple with electric field to apply a body force. But we know that this force is localized only in the EDL, which is of order few nanometers, and that is about 3 orders of magnitude smaller than the thickness of the channel. So you will see that this force is being applied only at the walls. So eventually you would get a velocity profile which is almost plug shaped. So this problem is similar to, let's say you have these two walls and these two walls are moving with some speed, so you would end up getting a plug flow. So it would appear that the flow is actually slipping on the walls because the thickness of this EDL is really small. But if you can somehow see into this EDL, eventually you could, and if there's a no slip boundary condition, you would see that within the EDL, the velocity goes back to 0 to satisfy the no slip boundary condition. So within the EDL you have this velocity gradient, but outside you have almost uniform flow. But because the thickness of EDL is really small, it appears that on the scale of the thickness of the channel that the flow is almost slipping on the walls, and you have a plug type velocity profile. So this phenomena is called electroosmosis. The another effect of electric field is that we have certainly positive and negative ions in the bulk solution, that is even outside the electric double layer, and under the effect of applied electric field these positive and negative ions also move. So if we apply electric field in this direction, positive ions would move along the electric field and the negative ions would move in the opposite direction. Okay, and that is just migration of these charged ions or particles under the effect of electric field, and this is called electrophoresis. So that means you can have both fluid flow and transport of mass transfer of ionic species by applying electric field. So the transport of fluid due to applied field is called electroosmosis, and the ion transport due to electric field is called electrophoresis. So we can now summarize. So what we have is that by applying an electric field we can have motion of charged particles or surface relative to a stationary liquid, okay, and that is called electrophoresis. So that means even if you have stationary liquid and you have a charged particle and you apply electric field, there would be a motion of that charged particle. That is called electrophoresis. Okay. Similarly, if you have a liquid which is present next to a charged surface and there are ions in the liquid, then by applying electric field you would have motion of liquid relative to the charged surface, and this is called electroosmosis. We can also have the opposite of electrophoresis. That means in this case we don't apply an electric field, but we have charged particles which are made to move in a stationary liquid, and that would result in formation or generation of an electrical potential. Okay, so motion of charged particles in a stationary fluid would lead to a potential difference or electric field, and that's called sedimentation potential. And application of electric field on a particle within a stationary liquid will lead to motion of particles, and that is called electrophoresis. So sedimentation potential is just the inverse of electrophoresis. Similarly, we have the inverse of electroosmosis, and that is called streaming potential. That means if we have a surface which is charged and we make an electrolyte solution flow over that surface, then that would lead to an electric field or a potential difference. This is called streaming potential, which is inverse of the electroosmotic flow. So these processes, electrophoresis, electroosmosis, sedimentation potential, streaming potential, are together termed as electrokinetic transport phenomena. So electrokinetic transport phenomena is actually a sub-branch of electrohydrodynamics, and this phenomena, electrokinetic transport phenomena, deals with those processes where there is a relative motion between a charged surface and the liquid. So we have the motion of a charged surface relative to the liquid in electrophoresis, electroosmosis, and even in sedimentation potential and streaming potential. And this sub-branch is called, of electrohydrodynamics, is called electrokinetics. Now let's look at a simple example of electrophoretic motion of a charged particle. So we can see how this particle behaves when we apply an electric field. Okay, so let's consider the case of a particle which has some charge Q and it is placed in an insulating liquid. So we are looking at this case of this particle submerged in an insulating liquid because if we have an electrolyte solution which can conduct current, in that case you will have positive and negative ions in the solution which would be attracted towards these particles, and then this charge on this particle would be screened, and that would complicate the physics. So we just want to look at a very simple case at the moment. So here we have this charged particle in an insulating liquid and we apply an external electric field E. So the force on this particle, the Coulomb force, on the particle would be Q times the electric field. So in general, we can consider electric field in any direction, so it is a vector. Now this is the force which is driving this particle, but when the particle tries to move, it acquires some speed, let's say u, relative to this stationary liquid, then it would experience a drag force. So let's say this is a spherical particle, and we know that the drag on a spherical particle is given by 6 pi times the dynamic viscosity of the liquid times the radius of the particle times the relative velocity, which is u, and this force would act in the opposite direction to the flow of the particle, so we have this negative sign. Now we can write the equation of motion. If we neglect Brownian motion for the moment, then the equation of motion would be m du by dt is equal to q times E, which is the Coulomb force acting on this particle, minus the drag force. Okay, now we can actually divide this whole equation by 6 pi eta r, and you will get m over 6 pi eta r du by dt is equal to q over 6 pi eta r times electric field minus u. Now this is the left hand side is actually the inertial term, and we can see that this inertial term is actually going to be really small. So the units of this coefficient over here, which is m over 6 pi eta r, is that of a time which is the relaxation time, and if you take the mass to be density of the particle times 4 pi r cube by 3, so you will see that if you compute this response time for this particle of a very small particle, it's going to be really small. So we can neglect these inertial terms, and this is something similar to what we saw in PIV, where we had small particles and they would respond immediately to an external flow. So similarly, a small particle would respond immediately to applied electric field. So eventually we would get that the speed of the particle under applied electric field is Q over 6 pi eta r. That means the steady state speed of the particle, which it attains instantaneously because it has a very fast response time, then it is proportional to the electric field. And this proportionality constant is called the electrophoretic mobility. Now we can see that the electrophoretic mobility, which is denoted by mu, depends on the charge and on the size of the particle, and of course the viscosity of the liquid. So if you have higher charge, then you would have higher mobility, and if you have larger size of particle and higher viscosity, you would have lower electrophoretic mobility. Now, although we have derived this relation for a particle in an insulating liquid, we can see experimentally and also derive that if we put this charged particle in a conducting liquid, let's say an electrolyte solution, even then we would get a similar dependence of this electrophoretic velocity of the particle, and this relation that U is proportional to the electric field generally holds, and the proportionality constant is called the mobility. And this relation holds for small ions and even larger particles, and for macromolecules, for example DNA, and also proteins. And it's obvious from a relation like this that higher the charge, the more would be the force acting on the particle, and that would lead to a higher electrophoretic velocity, and therefore the mobility would be higher. So now that we know that how we can model the motion of a charged particle under the applied electric field, in the next lecture we would see how we can model a concentration of a large collection of these ions or particles under the effect of an electric field, and we would also consider how this electric field gets coupled with the fluid flow and leads to this electrokinetic transport phenomena. So in the next lecture we will derive the governing equations for electrokinetic transport, and there we will see that this mobility is actually used to define the flux due to electromigration of these charged particles.